I'm Samara Irtaza, and today we are going to discuss flavonoids, their biosynthesis and functions. Today we are going to study its introduction, chemistry, classification, sources, biosynthesis of flavonoids, their functions, and role in plants. Flavonoids. They are a diverse group of phytonutrients, means plant chemicals, found in almost all fruits and vegetables. Along with carotenoids, they are responsible for the vivid colors in fruits and vegetables. Some of the best known flavonoids are quercetin and camphorol. Flavonoids represent diverse class of naturally occurring polyphenolic metabolites. Diverse means they are structurally different in nature. They have more than one phenolic group in structure. That is why are referred as polyphenolic in nature. They are found in either free form or glycosides, means they are attached to the sugar. More than 10,000 different flavonoids have been identified, and surely this number will increase, and they represent the third largest group of natural products. Secondary polyphenolic metabolites. Plant metabolites can be divided into two groups. Primary, which are involved in the nutrition and essential metabolic processes, for example carbohydrates, lipids and proteins, and secondary ones which have an important function in interaction between plant and its environment, such as pigments or defensive compounds. They are yellowish pigments as they are named from the Latin flavus, meaning yellow. Chemistry of flavonoids. There are 15 carbon atoms and their basic skeleton, which are arranged in the form carbon 6, carbon 3 and carbon 6, which means first 6 carbon atoms and the last 6 carbon atoms, they are mainly arranged in aromatic rings, and these both aromatic rings are connected with each other with the help of 3 carbon atoms, which may or may not be in the cyclic form. Classification. Classification of flavonoids is based on either position of phenyl ring B, degree of oxidation, or saturation present in C ring. For example, anthocyanins, flavonols, flavonol, proanthocyanidins, and chalcones. Broadly, there are 6 major classes of flavonoids: anthocyanins, chalcones, flavanones, flavones, flavonols, and isoflavonoids, which are further subdivided into several subclasses. And from here you can see their natural sources through which they are extracted. Mostly they are fruits, vegetables, medicinal plants and others. Biosynthesis of flavonoids. Here I have provided a basic overview so to help you understand the mechanism or biosynthesis. Comeryl-CoA, which is precursor of schemate pathway, and malonyl-CoA, which is a precursor of acetate pathway. They both condense together to form chalcone through chalcone synthase, which is further isomerized to flavanone through chalcone synthase isomerase. This flavanone, if undergoes hydroxylation, it produces flavanol, which is further oxidized to produce anthocyanidine. And of this flavanone, if it undergoes oxidation, then it produces flavone, which is further hydroxylized to form flavanol. Flavonoid biosynthesis follows the phenylpropane metabolic pathway, in which comeryl-CoA is formed from amino acid known as phenylalanine, which is mixed together with malonyl-CoA to form a group of substances known as chalcones. These are the backbones of every flavonoid and anthocyanin biosynthesis. This reaction is catalyzed by chalcone synthase that belongs to the family of polyketide synthase. This biosynthetic pathway has been characterized in Arabidopsis thaliana enzymes. Condensation leads to the production of colorless flavanone named naringenin, and oxidation of this compound by flavanone 3-hydroxylase yields dihydrocamphorol that can be hydroxylated by F3H or F35H to yield dihydroquercetin or dihydromycetine. Naringenin may be directly hydrolyzed by F3H and F35H to yield erodicitol and pentahydroxyflavone, which are further hydroxylated by F3H into dihydroxyquercetin and dihydroxymycetine. The three dihydroflavonols are converted to anthocyanidins, colored but unstable pigments, by two reactions catalyzed by DFR, dihydroflavonol reductase, and LDOX. DFR catalyzes a reduction of three dihydroflavonols into leucocyanidine, leucopelargonidine, and this one, which are further oxidized by LDOX into three anthocyanidine compounds. Cyanidine, it is red magenta anthocyanidine. If I talk about this one, it is of orange color, and delphinidine is purple mauve anthocyanidine. Last common step for the production of colored and stable compounds involves glycosylation by enzyme UFGT, which is UDP glucose flavonoid 3-O glucosyl transferase. Finally, cyanidine 3-glu and delphinidine, these both compounds, they can be further methylated by methyl transferases into pionidine and malvidine and pitunidine. Biological activities in plants. In plants, flavonoids cover a wide range of functions. Although they mainly act as yellow pigments in petals and leaves to attract pollinating insects, they might also appear as bluish pigments to receive certain wavelengths of light which permit plants to be aware of photoperiod. In plants, they play ecological importance in nature as they aid to pollination and seed dispersal. They have been known to synthesize in particular sites and are responsible for the color and aroma of flowers. And in fruits, as I have told earlier, they attract pollinators and growth and development of seedlings, which is an essential step in reproductive process. They protect plants from different biotic and abiotic stresses and act as unique UV filter functions. Flavonoids act as regulators of cellular cycle. Some of them are synthesized in plant fruits, and they have crucial roles in fighting infections caused by pathogenic fungi. Currently, there are about 6,000 flavonoids that contribute to colorful pigments of fruits, herbs, vegetables, and medicinal plants. Beneficial effects. As you can see, flavonoids have following effects. They act as anti-inflammatory, antiplatelet, scavenging free radicals, enzyme receptor system inhibition, anti-carcinogenic. They protect skin from UV light exposure, protect DNA from damage, strengthening of capillaries, protection against radiation, moistening, soothing, antiseptic, and other. Flavonoids have relevant pharmacological activities such as antioxidant, anti-inflammatory, anti-allergic, antibiotic, antidial, and against cancer. Some studies indicate that a diet rich in flavonoids can diminish the risk of cancer, but there are no significant statistics. As antioxidants, flavones and catechins seems to be the most powerful flavonoids for protecting the body against ROS, reactive oxygen species. And they also have anti-ulcer effect. Quercetin seems to play a very important role in prevention and treatment of peptic ulcer. It acts by promoting mucus secretion and thereby serve as a gastroprotective agent. Anti-atherosclerotic effect. Atherosclerosis, it is referred as hardening of arteries. Flavonoids seem to suppress inflammatory progression in artery wall. And they also have anti-inflammatory effect.